When I was small, I always wondered what my blood type is. My parents told me that I have A positive blood since both of them are blood A positive. When my parents informed me that my blood type was A positive, I felt a strange sense of pride. If A positive was the top grade in school, then surely A positive was also the most excellent type of blood. It didn't take long for me to recognize just how silly the feeling was and tamp it down. By the time I was in secondary school, I learned how to perform ABO blood grouping tests by using tau method. Oh no! What I realized is blood grouping tests actually show that I have O positive blood. But what are the other methods that we can use to determine what specific type of blood we have? There are a few methods that we can use in ABO blood grouping tests, such as slide or towel technique, tube technique, gel cut technique, as well as microplate technique. Well, slide or towel technique can be performed by using white towel or glass slide. If you are using glass slide to perform the test, we need to mark one side of the glass slide as A and another side as B. Then we put one drop of anti-sera A on the side marked as A and one drop of anti-sera B on the side marked as B. One drop of test blood sample is then added to each anti-sera and mixed by using a clean stick to look for agglutination reaction. A antigen will only agglutinate with anti-A, while B antigen will only agglutinate with anti-B. Group O blood will show no agglutination with either anti-A or anti-B, while Group AB blood will show agglutinate with both anti-A and anti-B. One thing that we must always bear in mind while using slide or towel technique to perform the test is the test can only be performed in emergency ABO grouping tests only, but not performed as a routine test. Well, the advantage of using this method is it can be used in emergency and blood camps for preliminary grouping, and it is easy to be performed and very quick to get the results. However, it is not reliable for weak reactions as negative results cannot be checked microscopically, and serum testing cannot be performed by using this method. The test mixture also tends to dry too fast, and drying can cause aggregation of cells which can be misinterpreted as agglutination. This method also less sensitive than tube technique. This test is performed by using forward grouping and reverse grouping. Forward grouping is used to test the patient's red blood cell by using known anti-A, anti-B, anti-AB, and anti-D reagent. Reverse grouping is used to demonstrate the corresponding presence or absence of anti-A and anti-B in the patient's serum or plasma by using known A cell, B cell, and O cell reagent. The procedure for cell and serum grouping is described separately, but in all samples, both the procedures should be done simultaneously and the results cross-checked. The presence of agglutination is a positive result and can be graded as 1+, 2+, 3+, or 4+. If a smooth cell suspension appears after the button is resuspended, it is a negative result. There are a few advantages by using tube method, such as it is easy to be performed, accurate and more sensitive than slide technique. It allows for fairly long incubation without drying up the tube content. The centrifugation used enhances the reaction, and hence even weak antigens or antibodies can be detected. It uses smaller quantity of reagent and is clean and more hygienic. However, it requires extensive labeling and quite subjective in manipulation of tubes. To prevent false positive or false negative of the results, we must always use thoroughly clean test tube for the test. While reading the results, shake very gently to dislodge the button. And we must always use correct speed and time centrifugation to avoid erroneous results. In gel cut technique for ABO blood grouping test, microtubes containing sulfadex gel prepared in a buffer such as low ionic strength saline or saline unavailable. In the cards used for blood grouping, red cell specific antisera and a preservative are also added to the gel at the time of manufacture. Each card contains 6 such microtubes. There is 1 negative control, 2 with neutral gel served for reverse grouping with A and B cells, and 3 for forward grouping by using specific antisera. Positive results is shown when the agglutinated cells form a red cell line on the surface of gel or agglutinates dispersed in gel. While in negative results, we will see a compact button of cells will sink to the bottom of the microtubes. The advantages of using gel cut technique is the reaction produced is relatively clear and stable for at least 48 hours. Unlike in tube technique, the washing step could be omitted when using gel cut technique. This would in turn slightly shorten the time required for the results to be produced. Hence, technical factors such as improper washing of red cells that may cause false negative results can be avoided. By using gel cut technique, a lower volume of sample is required as compared to the tube technique. Gel cut technique is standardized and relatively easy to be performed, and it is less subjective in interpretation. But the bad thing is presence of clots, fibrin strand, or particulate materials that entrap cells and prevent them from moving through the gel may be misinterpreted as positive results. Using gel cuts would cost more than using tube technique. It also requires acquisition of some dedicated equipment and some specialized training for their use. And by using this method, it will cause loss of traditional skill. Besides the techniques mentioned before, there is also one more technique, which is microplate technique. Microplate technique is performed by using microplate, which consists of a plastic tray with 96 wells. There are 3 types of microplate, which are V-well, U-well, and flat bottom. Usually, V-tap well is preferred because of ease of suspension for red cell serological work, whereas U-tap well is usually for ELISA technology. The blood are arranged in serial order in the well. Each microplate has 8 wells vertically. In the first 3 wells, put 1 drop of diluted anti-A, anti-B, and anti-AB respectively. In the next 3 wells, we put 1 drop of A cell, B cell, and O cell. The remaining 2 wells can be used for RH typing. Then, gently tape the plate and incubate at room temperature for 1 hour. If a microplate centrifuge is available, centrifuge at 200 grams for 1 minute after 15 minutes incubation. After that, resuspend the red cells using a microplate shaker or manually briefly. In positive results, we will see a carpet of red cells which line the bottom of the well. Whereas in negative results, a compact button with smooth edges which streams when the plate is tilted will be seen. The advantage of using microplate technique is It uses only small volume and low concentration of sera and red cell, thus making it more cost-effective. It is easier to handle microplate in place of 96 tubes. Large number of samples can be processed at the same time. There is economy of space and time. The technique can be automated, which help to reduce the reading and transcription error. However, this technique requires considerable technical expertise. I am sure that now you have learned how to perform the ABO blood grouping test as well as each technique's advantage and disadvantage.